Welcome everyone. Today we are going to learn about resting membrane potential in neurons. In my previous video, action potential, I have mentioned that resting membrane potential is the baseline of action potential from which action potential is generated. Now, if we insert a microelectrode inside a unstimulated neurons, we will get a value of membrane potential that is called resting membrane potential. And the value differs, ranges between minus 40 to minus 90 millivolt in different species. Like in case of squid neurons, it is minus 70 millivolt, and in case of human neurons, it is minus 40 millivolts. So it differs or actually ranges between minus 40 to minus 90 millivolt. Now, why there is a resting membrane potential at all in an unexcited neurons? The answer is due to the movement of ions. So ion movement creates this resting membrane potential. This is the first answer. Now the question is why there is movement of ions. For that questions we will talk about two forces, that is the chemical force and electric force. So for the movement of ions there is two force works, that is chemical and electric. Now the chemical force is called ion concentration gradient. Now another question comes, why there is ion concentration gradient? Now the answer is due to the presence of ATP driven pump in the plasma membrane. Now what is ion concentration gradient? Ion concentration gradient means there is a difference in ion concentration in the inside and outside of a cell. Suppose this is the plasma membrane that surrounds the action of a neuron, then the inside, the cytosol, and the outside, if there is any difference in ion concentration, we will call it ion concentration gradient. Now in the plasma membrane there are many different type of ATP driven pumps are present. These pumps are active transporter because they break down ATP, hydrolyze ATP to pump ions to generate ion concentration gradient. Now in this case I have taken example of just a single ATP driven pump like sodium potassium pump. Now let's see how this pump generate the ion concentration gradient of sodium and potassium. See, this pump actually export 3 sodium out of the cell and import 2 potassium. Now, as because this pump export 3 sodium, the external concentration of sodium is higher than internal sodium concentration. Like sodium concentration in outside is 144 millimolar and inside is 12 millimolar in all vertebrate cells. Now, as because they import potassium, so the concentration of potassium outside is lower, that is only 5 millimolar, and inside is higher because 155 millimolar. Now in the plasma membrane, as I have mentioned, there are different type of ATP driven pumps are present. So for chlorine ions also there is another ATP driven pumps present. I have not drawn here because why I am mentioning potassium, sodium and chlorine, because for animal cells these 3 ions, sodium, potassium, chlorine, actually responsible, are major contributors for the resting membrane potential and membrane potential. Okay, so there is another ATP driven pump present that actually creates the concentration gradient of chlorine. Like in case of vertebrate, the outside concentration of chlorine is 110 millimolar higher than inside chlorine, that is 4 to 30 millimolar. Now this is the chemical force that helps in the movement of ions. So ion concentration gradient is first point we have to understand. And why there is ion concentration gradient? Because of this presence of ATP driven pump that hydrolyze ATP to generate this ion concentration gradient. So I am talking about these 3 ions only. Now basically the ion movement occurs through ion channels, not through this pump, ion channels. Now ion channels move ions from higher to lower concentration. The ATP driven pump creates the ion concentration gradient, the chemical force, and the ion channels utilize the force. They move ions from high to lower concentration. Now let's suppose in an unexcited condition, suppose these 3 channels are present in the external membrane, then if these channels are open, what will happen? As because potassium concentration is inside is higher than outside, potassium will come out. As because sodium concentration is higher outside, so sodium will come inside. And as because chlorine is concentration is higher outside, so chlorine will come inside. So through the channels, ions move from high to lower concentration, and that difference is created by the ATP driven pump. We call it ion concentration gradient or chemical force. This is the first point we have to understand at first. Now, let me explain the second point, that is the electrical force. Generally, basically, this electric force is the opposing force of chemical force or ion concentration gradient. Now, what will happen if these channels are open? What will happen? There will be a membrane potential. Why? Membrane potential means voltage gradient also. Like the ion concentration gradient, membrane potential is also called voltage gradient. That means there will be a difference in voltage inside and outside of cells. Now, why there is membrane potential will generate? Because these ions carry charge. Like potassium is positively charged, sodium positive charge, and chlorine is negative charge. That's why when these ions move, they also create a membrane potential or voltage difference. That's why we will get a value that is, in case of squeak neurons, as I mentioned, it is minus 70 millivolt. Why I am mentioning here? Because Alan Hodgkin and Bernard Koch actually proved which ion is actually responsible for, particularly responsible for resting membrane potential, because these three ions actually helps in the generation of membrane potential. But for resting membrane potential, one ions will be very important, and they discovered this fact in 1949 from Cambridge University. They took the help of squid giant neurons and they got a value that is approximately minus 70 millivolt in physiologic condition. That means in the intact organism, squid, they got the value minus 70 millivolt. They just simply insert a microelectrode inside the giant axon and they put another microelectrode outside. They got this value minus 70 millivolt. So this is the membrane potential or voltage gradient because of this movement of these ion channels. Now, this Alan Hodgkin and Bernard Koch want to know which ions is responsible for the generation of resting membrane potential. They perform a very simple experiment. What they do, they actually taken out the giant action from the squid to an external environment filled with liquid, and in that liquid they actually put these ions. At first they actually take the ions just like the physiological concentrations, like sodium is 144 millimolar, potassium 5 millimolar, and chlorine 110 millimolar. When they take this ion concentration just like the physiological conditions, they got the value of minus 70 millivolt. See, this is the experimental conditions. They also got the physiologic value. Now they actually start to change the concentration of outside ions, because to change internal concentration of these ions is very difficult. We can easily change the concentration of sodium, potassium, and chlorine outside. So as because in the physiologic conditions the sodium concentration is higher, so they try to lower the sodium concentration to see that if there is any change occur in the resting membrane potential. They try to increase the potassium concentration because in physiologic conditions the potassium is lowered in concentration normally, and they also try to lower the chlorine concentration outside external to see if there is any change occur in the resting membrane potential. And what they found clearly shows that if we change the sodium and chlorine concentration or decrease the sodium chlorine concentration, very little voltage changes occur. That means the sodium and chlorine has a little effect on resting membrane potential, that is minus 70 millivolt. But when they change or increase the potassium concentration, they find that a significant change occurs in the value. So they pinpoint that this potassium ion is responsible for this generation of resting membrane potential. So what they did, they actually increased the potassium concentration. Now, if we increase the potassium concentration, what will happen? The ion concentration gradient will be lower, because I have mentioned the ion concentration gradient is the difference in ion concentration. So if we increase the ion concentration, potassium concentration outside, what will happen? The ion concentration gradient, the chemical force will be lower. As because there will be low chemical force or low ion concentration gradient, so low amount of potassium will move, and that's why the membrane potential will drop. So the membrane potential will be minus 60 millivolt, it could be minus 50 millivolt or minus 40 millivolt. And in this way, as we will increase the potassium concentration outside, the membrane potential will fall. And they found the exact same thing. That means the concentration of outside potassium is linearly related with the voltage of this resting membrane potential. Now they did another thing, that they actually increase the potassium concentration outside just like the inside concentration, 155 millimolar. then what will happen? There will be no difference inside and outside of the potassium. So let's suppose we take the potassium concentration 155 millimolar, increase the potassium concentration. So there is no ion concentration gradient, so there is no chemical force. So what will happen? There will be no potassium movement. So what will happen if there is no potassium movement? The resting membrane potential should be 0 millivolt. And they find that exactly when we increases the potassium concentration outside, just like 155 millimolar, we got a resting membrane potential that is 0 millivolt. So that clearly shows that potassium ions is important. So potassium channels are important. Now subsequent research also proved that many potassium channels remain open during the resting condition or unexcited conditions, and very few sodium and very few chlorine channels are open during unexcited conditions. Now these channels, potassium channels, are called non-gated potassium channels. Remember this, non-gated. Why? Because this channels opening and closing does not depend on voltage or ligand or mechanical stress. Because in the plasma membrane there are different type of voltage channels are also present called gated channels that are regulated by voltage, ligands or mechanical stress. But these channels are non-gated. That means they does not depend on external force. So they remains open. That's why these channels is also called, these potassium channels are also called leaky channel. Leaky potassium channels. Okay, remember this, leaky potassium channels, as because they remain open during the unstimulated conditions, and that's why they generate the resting membrane potential. Now this family of these leaky potassium channels also called KIR, that means potassium inward rectifier, this family. So this leaky channel falls under the potassium inward rectifier family. Now when depolarization occurs, that means when action potential occurs, magnesium ions actually block the passageway of the potassium ions. And when the repolarization occurs and then it comes into the resting membrane potential, then the magnesium actually come into the cytosol. So from the cytosol the magnesium enters and it blocks the passage of the potassium ions, and then it actually comes out when the repolarization occurs, when the action potential stops. So magnesium actually block the potassium inward rectifier channel or leaky potassium channel, or we call it non-gated potassium channels. So from Alan Hodgkin and Bernard Koch experiment in 1949 from the Cambridge University, we got the value minus 70 millivolt, and we know that potassium ions or potassium channels are important for the generation of resting membrane potential. These two forces, ion concentration gradient and the membrane potential, that means the chemical force and electric force, if we combine these two forces, we will call it electrochemical gradient. Okay, now as I have mentioned, these two forces actually are opposing force. Chemical force or ion concentration gradients try to move the ions, but at certain membrane potential the movement actually stops. Why? Because see, if potassium ions move outside, what will happen? More potassium ions will be present outside and less potassium ions will be inside. That's why there will be more negative ions will be present inside, so there will be negative charge, and outside will be more positive charge. So as more and more positive ions or potassium ions will accumulate outside, that accumulated positive ions will repel the further movement of potassium ions. So at a particular membrane potential, this ion concentration gradient or chemical force is opposed. So at certain membrane potential, the chemical force actually just balance the membrane potential. So at that time, the electrochemical gradient or this combination of these two forces will become zero value, and in that the membrane potential at which this electrochemical gradient becomes zero, we call it equilibrium potential. Now as I have mentioned that mainly potassium ions are responsible for the generation of resting membrane potential. So this value minus 70 millivolt actually indicates the equilibrium potential of the potassium. So we want to check, because Alan Hodgkin and Bernard Koch actually performed experiment, so they got this value. So we will actually want to know exactly that the value of equilibrium potential for the potassium will be minus 70 millivolt or not by one equation called Neurons equations. So by Nernst equations, we will validate that whether really this potassium is responsible for the generation of resting membrane potential.